Yeah, so welcome everyone to our first SynoProtein webinar. The title of our first webinar is Forest Residues to Alternative Protein and Bio-Based Carbon Materials. And first of all, thanks for signing up and for your interest. While people are still coming in, I would just like to give you a quick overview of our agenda today. So we will, in a second, I will hand over to Long Ling from via Environmental Solutions, who will give us an introduction to the SynoProtein project about carbon capture from syngas to single-cell protein and its use as fish feed ingredient. After that, Hege von Midtgaard from Bergene Holm in Norway will talk about finding the best purpose for every part of timber resources. Next in our agenda will be Jan Hendrik Klüt from SINTEF in Norway, who is going to talk about the integration of biomass, flow pyrolysis with biochar cracking for carbon capture and utilization applications. And after that, we will hear about conversion of pyrolysis gas into biohydrogen via gas fermentation from Konstantinos Chandolias from RISE Research Institute in Sweden. And last but not least, Yao Li from Technical University in Denmark is going to talk about gas fermentation for single-cell protein production. And in the end of the webinar, we will have a short section for questions and answers, discussion with all the speakers. And you're very welcome to post any questions that arise during the presentations to our Q&A section, and we will address them then later on. I want to just give a note that this is a very industry-driven project, so the partners can, of course, not share too many details about all the technologies, but you're very welcome to post your questions and we can discuss together in the end of the webinar. And with that, I'm happy to hand over to Long, and she will give you an introduction to our project.
Thank you, Christina, for the introduction. And I'm going to share my slides. So hope everyone see my full screen presentation.
Yes, looks good.
Perfect. Yeah, thank you and welcome everyone to our first webinar on SynoProtein. And my name is Long Ling. I'm the Chief Science Officer at Y Environmental Solutions. And first of all, I would like to mention that SynoProtein Project is a industry-driven project again, and is supported by Circular Bio-Based Europe. It's a partnership between Bio-Based Industries Consortium and EU. Then, first of all, before jumping into the project, I'd like to introduce a little bit who we are at Y Environmental Solutions. Our vision is to make a sustainable future by replacing fossil carbons with biocarbons. And we are a SME founded in 2017 in Tønsberg, Norway. Tønsberg is in the Vestfold area, like southeast of Norway, if you don't know much. And it's quite close to the capital, Oslo. We also have a branch office in Suzhou, China, and another branch office in Poland. Our main sector is in the bio-based materials. We are also a technology provider and have our pilot plants. Now, our main products is the metallurgical biocarbons. We have developed targeted for different industry needs. For example, steel industry or silicon industry, ferrosilicon, and aluminum industry as well. These biocarbon products can be directly replace fossil coal or coke in their process to reduce the carbon emission. Then our products intend to be used by our customers without new investment on the infrastructure. And some products are under development, for example, like premium biocarbon for the battery materials are still under development. Then, in addition to the main product biocarbon at Y, we also have syngas as the byproduct. Then, how to valorize this syngas byproduct? That's the idea behind the SynoProtein. So we came up with this SynoProtein project, carbon capture from syngas to single-cell protein and use as fish feed ingredient. The project received 5 million grant from the CBE-JU, and also it lasts for 54 months. It started last year. It's going to end in 2028. For the project-wise, the feedstock can be the forest residues or syngas, depending on the processes involved. Then our projects aim to develop and demonstrate in the prototype pilot a novel carbon negative process that can convert forest residues or syngas into single-cell protein for fish feed, and as well as producing biocarbon for the animal feed. Then by transforming this low-value byproducts from sawmills and into high-value bioproducts, SynoProtein can provide a sustainable protein alternative to meet future demands. We are a consortium with 11 partners from 4 different European countries, like Norway, Sweden, Denmark, and Germany. We are the project lead, Y Environmental Solutions, and we are also the only SME in the consortium. And we also have 8 research institutes and 2 large companies in the consortium. The 2 large companies are the Bergenholm, and which is the 2nd largest sawmill in Norway, is the feedstock supplier project, and Skretting is the global lead in aquaculture feed is the product off-taker in the project. So our consortium integrates the whole value chain from the feedstock supply to the product offtake. And with the great consortium, the problem we are trying to address is the increasing protein demand. As we know, the protein demand is increasing and will double by 2050. And accordingly, the animal feed will increase as well. But there is a huge gap. The current production of the animal feed that cannot meet this increasing demand. And also the traditional production of the animal feed might have different challenges, like the deforestation, and also due to the limited marine resources, it cannot be sustainable. So the whole industry is looking for sustainable protein alternatives to meet this increasing demand. Then we propose the SynoProtein from syngas to protein. Then the syngas have both the carbon and energy, can be used as the feedstock for the protein production. The syngas is also abundant and cheap byproduct from forest residues. Then the whole process has very high CCUS potential. Then the production is also fast and has high protein content and low land and water requirement compared to traditional production, for example, soybean cultivation. And also compared to the state of art. On the feedstock side, we know the sawmills, usually about half of the wood will end up as a sawmill byproduct. And this part, how to use the byproduct, the state of art process is either neutral process and also from the aquafeed side. The traditional production of the aquafeed can be carbon positive, and also is due to the limited marine resources. Of this state-of-art technologies, our SynoProtein solutions can be carbon negative by integrating the production of both single-cell protein and the biocarbon. The biocarbon can also reduce the carbon emissions in the cattle farming as well. So by the whole process, SynoProtein can help achieve different sustainable development goals under EU. So like emission reduction, industry innovation, sustainable production, and also protection of water and marine resources. And with this, the SynoProtein can create values for different parties. And the first of all is recycling of the low value byproduct into high value bioproducts. And also it can help diversify the feed ingredient source by offering sustainable protein alternatives. And with this production of both single-cell protein and biocarbon, it can largely contribute to the overall profitability. And with this, it can unlock the revenues for the forest residue feedstock supplier. And use the syngas with the renewable carbon and the hydrogen source. It can also achieve cost and carbon savings compared to other single-cell protein production technologies. And with this technology, we see a huge market opportunity for the product. The whole market represented by the global animal feed protein. We would like to target first is the aquafeed protein because of the market interest and also the good collaborators in this project. But the whole market is driven by the feed sustainability charter, which is the climate neutral aquaculture and livestock production through feed. So in the end, there is a huge market developing for this sustainable feed. In order to seize the market, we also develop a milestone for the market entry, a very ambitious roadmap. Then this roadmap is together with the financial development, technology, and product development. First, let's take a look at what we already achieved. The SynoProtein Consortium already formed in 2022, and also the project started in 2023. And in order to reach the market entry by 2030, we have planned step by step to reach different milestones of production. First, have to achieve the prototype pilot, which is also the goal in this SynoProtein project under the CBE-JU support. And another further is we have to scale up to the pilot, and then further scale up to the full-scale demo plan. And in parallel to that, from the product side, we need to have the product test and product certification, and also the offtake agreement, and so on. With this very ambitious roadmap, we have a very good team behind to support the project. And you can see on the left side is the our staff from the Y Environmental Solutions, and we have very professional team to cover various disciplines for this SynoProtein. And also we have the top experts from all other 10 organizations in the whole consortium. And we are so happy to have so many top leaders in their field in our project that, as we mentioned, covers from the feedstock supply to the product offtake. And in addition, we also have the regulatory compliance expert and the ESG assessment and technology and engineering in the middle as well. And not only we have a great team, we also have a good plan to execute the project. The work packages mainly includes 5 technical work packages and also 3 communication and management work packages. So first is the pyrolysis and syngas conditioning, led by the SynTEF, which you will hear in a minute. And then the second is the syngas fermentation and single-cell protein, which is led by DTU. And you will hear the report from RISE and DTU today as well. Then the number 3 is the pilot demo, which is led by Y. Then NOFEMA is the Feed Research Institute in Norway. Environmental assessment led by NOSIS. The communication led by HB, it's the University of Borås in Sweden. And then is the business plan development led by the KIMA, which is the organization of this webinar as well. Is a research institute in Germany. And finally, the project management. And you will, after me, you will also hear the feedstock from the feedstock side, which is Bergenholm is the feedstock supplier in the project again. So that's the overall project introduction. Thank you for your attention, and I'll happy to have questions at the end. Thank you. So I'll stop sharing.
Thank you very much, Long, for this nice introduction to the project. And I will now hand over to Higava Midgard for the next presentation.
Yes, thank you so much. I hope you all can hear me. I'll just try and see if you get the right view on the presentation. I'll try another like this. Can you see like the full screen now, everyone?
Yes, it looks good.
That's good. Okay, thank you so much for attending. I'm going to talk a bit about our company and how our mission to use the timber resource in the best possible manner. And not just the timber resource, but see the possibilities like Long Ling have explained in every process that we do. So this is a short agenda of what I'm going to lead you through, and of course I'll be here in the end to ask questions if there are. So this is our company. We are a bit more than 450 colleagues, and to see the amount of timber that we every year buy, it's approximately 25% of the Norwegian sawn timber. So we are, like also has been said, the second largest. But that said, 1 million cubic meters every year in Norway we harvest in total, both the mass timber and the sawn timber, approximately 12,000 millions. So since we are sawmills and planing factories, we buy the highest quality timber. And I'm here in a bit south on the map. Our headquarters is quite close to Tønsberg It's in Larvik, and you can see the Norwegian map on the right side where we have our factories. And one of the largest one, at least the one we have invested mostly in and still to expand, is the one closest to Tønsberg. And biosyn, maybe some of you know that, we have for many years wanted to make the most sustainable use of all our both products and byproducts. So of course, biocarbon, biofuel, to find more sustainable purposes than just direct burning. So this is our strategy house that we have been working towards for quite a few years now. And you can see in the bottom, and I see that's in Norwegian, but maybe some of you know the UN Sustainable Goals and can sort of see the English translation. It's our sort of fundament to have that in everything we do. And of course, to be the best choice when building in wood, but in these times, even more important to use the whole resource, not just the building. So here we are, and one of our main targets in sustainability direction, that is to always reduce waste, energy, fuel, noise emissions, and the total CO2 emissions per unit produced. So this is, of course, one important thing for us, that innovation in both not just the biocarbon, but even the use of the syngas. So, and as the flag or the sign says on the bottom of the logo, it's important for us to not greenwash and just talk about how great the material and the photosynthesis, that's the work for us. Here you can see the factories and also the headquarters in the bottom right corner where I sit, quite close to Tønsberg. The factory in the middle is the first factory and the smallest one. And then we have some quite large units in the south of Norway. We have for many years been doing a lot of innovation for this is our main product. Or everything you can need for building a house or a wooden deck. So for us, like also Long Ling said, this is our main products, and we have approximately 50% of the log is byproducts. And here you can see for many years it's been quite challenging to find customers for it. And now in these sort of new times and energy crisis, it's not a challenge anymore, but the challenge is to find the best use for it. So this is just one example, and it's also it takes up a lot of space in the factories. So our normal value chain, this is what we normally work with. It's of course to get the raw material from the woods, and we do calculate every log that comes into us, and approximately 50 to 70 kilometers is the travel distance from the forest to our factories. So we have what you can call short traveled timber. And all our factories are certified as Miljøfyrtårn. It's qualified the same as EMAS or ISO 14001. So we have good environmental control in all the factories. And we have also been reporting with reference to the GRI for a few years. And all our main products, the ones you saw with the logos, the cladding and the timber that goes into buildings, we all have a life cycle analysis on all the products, because we need that for the customers, which are more and more environmentally concerned. They want to have green certifications or other, like you see the logos in the cities. And this part of the value chain, the sawmills, have always had a lot of regulations and control systems for having the right quality and standards for the whole production. This shows the amount of the two main sort of raw material that you can get from our factories. It's mainly spruce, but it's also pine, and we can see more increasing pine the latest years. And of course, we are working a lot with the forest to get the right length and dimension to get the best use of every log. And I think this is maybe the most important slide for now. That almost all Norwegian forests are PFC certified, because if you're going to make the most sustainable fish protein, we need the most sustainable raw material. And all our factories have 2 certifications, both PFC and also FSC. And it's certification, it's a chain of custody that every part in the value chain need a certification so we can trace where the log comes from. Yes, it probably says this slide says the same, but I think, as you know, more and more concerns about more than just the carbon and the climate is it's in what we are concerned about, so that we are aware of that the forest and the raw material we use is much more than tree and logs. It's also a place for different species and for us human beings to have free time and to get our health and a lot of feelings is sort of linked to the woods and to the trees. So in our work, we always think about who cares about our production. And like a few years ago, it was always the climate and the reduction of the CO2 emissions. But now we are also trying to calculate how we affect nature and the use of space. So I think that's an important thing to be aware of. This we have talked about. This is a bit optimistic drawing of how we can make wooden products for buildings out of a log. It is approximately 50% that goes to other customers, and some of you can see here. So in detail, from one log, we get approximately 10% is the bark, and that is counted extra from the what's inside of the bark. So we have a lot of bark, and that's mainly energy use, but also a bit gardening. And then like wood has a lot of water, so we have a bit of shrinkage, and everything else that doesn't go into the building products, like we have here 55%, is our byproducts, like the syngas is the byproduct from biocarbon. And we need a lot of energy for our production. We need to heat and to dry out the material, the building products. So it's also a really good like industrial symbiosis, or a way of not losing energy and potential in the process. So now I'm going into, since this was fish, some need of ingredients for protein for fish, it's just interesting also. I'm not sure if this process can also use wooden waste, but in Norway we have a lot of wooden waste. It's twice as much as the food waste. And I think it's just so sad. Now we are soon going into September. So in 1 month, it's the International Day of Awareness of Food Loss and Waste Reduction. And we don't really have any leftover dinners. And also the byproducts we have is often seen as waste, and it's just too good feedstock to not use for what we are trying for here. So thank you.
Yeah, thank you very much, Inge, for this nice insight into Bergina home. And with that, I'm handing over to Henry, who will give the next presentation.
Hi everyone, I hope you can hear me well and see my screen now, which should soon go to full screen. Everything looking okay?
Perfect, yes.
Okay, thanks for the introduction, Christina, and thanks everyone for joining the webinar today. So I'm Jan Hendrik, or Henrik Litte. I'm a research scientist at Syntef Industry, based here in Trondheim, Norway. And I'll be speaking about today about the integration of biomass low pyrolysis with biochar tar cracking for carbon capture and utilization applications, which kind of covers the work in work package 1, where I'm the work package leader. And so the work that I'll be presenting today is kind of done in collaboration between me and my colleagues Nicolas Canabara at Syntef Industry, our colleagues at Syntef Energy, a different Syntef institute, Liang Wang and Ask Listener, and then also project partners, Wire Environmental Solution, Rantau and Longlin. Okay, so I'll just start with a very broad introduction about what we're trying to do in this part of the project. But this is all about trying to exploit the synergies between biochar production and the utilization of the biochar or the pyrolysis gas from the biochar production process. So just to start to speak about what biochar is, so you can see an example of a typical biochar on the top right. And so biochar is a carbon-rich residue that remains after thermal decomposition of biomass, and it has a lot of potential applications. So one of them is as a more sustainable fuel to replace fossil fuels, for example. It can also replace fossil coal as a reductant in many metallurgical processes. It has applications as a soil enhancer, which is a way to store CO2 captured from the atmosphere. It has many applications as an adsorbent, and then also one that we'll be exploring in the project is applications as an animal feed supplement. So in the bottom right, you can see some very simplified schematic of a typical biochar production process. We can see that along with the biochar, you typically also produce a pyrolysis gas as a byproduct. And this pyrolysis gas typically is quite energy rich. So in the most common configuration, you would add air to combust the pyrolysis gas, and then you would be able to recirculate some of the heat produced to the pyrolysis process, where you use this as input to the process. But then the remainder of the energy produced would be used for either as heat for example housing, or to produce electricity. But so it's important to note that these are relatively low value uses of the valuable components in pyrolysis gas. So it's quite interesting to look at different processes where one would be able to produce high value products from this pyrolysis gas. Another important thing to note is that if you typically combust the pyrolysis gas, you emit the carbon that's part of this as CO2 emissions. So it's also interesting to look at possibilities of integrating this carbon into a byproduct, where it could, for example, displace CO2 emissions from other processes that are used to typically produce that kind of product. So speaking a bit more about the pyrolysis gas, so these gases typically contains non-condensable components such as hydrogen, carbon monoxide, carbon dioxide, methane, and other lighter hydrocarbons. It also typically contains a sizable fraction of water as steam. And then it contains condensable tars, which are typically defined as larger hydrocarbons with a boiling point above around 80 degrees Celsius. So that means that if you cool down the tars to room temperature, it would condense into bio-oil, as shown in this picture on the right. So the tar components in pyrolysis gas are typically very complex, and there's a large number of them. So in many processes, to be able to utilize this gas efficiently, it's simplest to break down the complex tar molecules to simpler and smaller molecules, such as hydrogen, carbon monoxide, CO2. And this would typically be done with metallic catalysts. For example, nickel is quite a good catalyst for this, which is used to reform the tars. So that means you add steam that partakes in the reaction. So the advantage of using metallic catalysts for this purpose is that they're typically very active for the breaking down these tar components. But the downsides are, depending on what metal you use, they can be expensive, rare, or in the case of nickel, toxic with some quite bad potential health effects. And then also they can tend to deactivate quite quickly due to solid carbon formation on the metallic nanoparticles, which then either leads to an additional cost because you have to replace your catalyst more frequently, or it can mean that you have to regenerate your catalyst by burning off the carbon, which typically leads to additional CO2 emissions. So a quite interesting possibility is to use the biochar from the pyrolysis process as a catalyst, because many studies in literature have shown that biochar is quite an active catalyst for reforming of tars. And this is due to several reasons. So one of them is the high porosity and surface area of biochar, where you can see some examples from scanning electron microscopy images on the right, which shows the pore structure and the high surface area of such biochars. Additionally, naturally occurring in the biochar is typically small fractures of alkali and alkali earth metal species that can act as catalysts. So here's an example showing some calcium on a biochar, and this can help to catalyze the tar cracking reactions as well. And then lastly, on the surface of the char are also some functional groups present. This shows an example of some functional groups detected using FTIR on biochar, and this can also further help the tar reforming reactions. So based on this, then biochar has some nice advantages compared to metallic catalysts. So one is the relatively low cost. Biochar can be sourced from feedstocks that are safe, abundant, and more environmentally friendly. And then importantly, the biochar is produced directly in the pyrolysis process. So you can see that there can be some nice synergistic integrations into the biochar production process. And then lastly, if you perform this under the right conditions, you can see your spent catalyst as a product instead of just having to see this as an additional cost where you have to periodically replace your catalyst. So based on this, we want to look at the following process in the SynoProtein project. The feedstock will be a byproduct from the sawmill industry, which can undergo some pre-treatment before being dried and being fed into the pyrolysis reactor. And as I mentioned, then the products are the biochar and the pyrolysis gas. The pyrolysis gas will be fed to a tar cracking reactor where the tars are broken down into smaller molecules. For this, you have to add some energy for these reactions, and here we want to look at using electrical heating to avoid having to combust a part of your biomass or your pyrolysis gas, which typically increases the CO2 emissions from the process. Additionally, from the produced biochar, you can split off a part of the stream, and we'll look at pre-treating this biochar to make them more active as catalyst, and then this catalyst can be added to the tar cracking reactor, and the spent catalyst can be taken from the process as an additional product. So after the tar cracking reactor, it has to be cooled and some heat can be recovered from the process. And then steam is added to the process for what is called the water shift gas shift reaction, which shifts your carbon monoxide coming from the tar cracker to CO2 and more hydrogen. After that, you can recover some additional heat and cool down the stream, and then condense out your water and any remaining tars that you have, leaving you with a syngas that contains mostly hydrogen, carbon dioxide, and then small amounts of methane and carbon monoxide. And this can then be utilized for single-cell protein production or other potential uses, which the other project partners will discuss more in their presentations. Okay, so then I can just as a bit of an appetizer of what's coming in the project, show some very preliminary results from the project that are first steps to deploying biochar as a tar reforming catalyst in a SynoProtein process. So the first part of the work is done by our partners at Environmental Solutions. And so they have, so far in the project, looked at producing biochar from different feedstocks. So this has been done with 2 different reactions. The one is a batch rotary tube furnace on the left, and the other one a continuous pilot rotary kiln. And they've also looked at producing biochar from different feedstocks. So for wood chips, they've looked at birch, pines, and spruce. And they've also looked then, in addition to at spruce sawdust. So in each row, you can see the feedstock and then the resulting biochar at the bottom. So some initial conclusions from this work. So the biochar compositions were quite similar for pine and spruce, but a bit different for birch, which is the only hardwood considered in the study. And specifically, a higher ash content was found, and this, for the use of the biochar as a catalyst, can be quite interesting because the ash typically contains these metallic components that I spoke about, which can act as catalysts in a process. Additionally, Wilde looked at using spruce from different sources inside Norway, but found that the growing location of the wood did not have a significant effect on the composition of the biochar that is produced. And then lastly, it was found in these results that the biochar from the spruce sawdust contained more fixed carbon and ash than the wood chips. So this might be related to the particle size of the sawdust that is quite a bit finer, but it's most likely because these experiments were done in the 2 different reactors, so the different operating conditions quite likely leads to different properties of the biochar. So in the rest of the project, we'll look at testing these biochars as catalysts, both as powders or pellets. And then specifically the next part of the work that I'll cover is our colleagues at Center for Energy is also looking at upgrading these biochars produced to make them more active as catalysts. So in the work they've done so far was done with using biochar pellets in a laboratory fixed bed reactor, where you can see a schematic of the setup here at the bottom. And this is done by adding carbon dioxide to the process, which then through the boudoir reaction gasifies your carbon to produce carbon monoxide. And by consuming the solid carbon, you produce more pores and open up the pore volume and increase the surface area for the reactions. So some preliminary results that found that, as expected, as you increase the reaction temperature for the same reaction time, you get more wait loss from the project, so you open up more pore space. And then additionally, the effect of the activation on the morphology of the pellet was quite clearly seen in SEM images. So here on the right is an example comparing the original biochar pellet, the surface, with the activated one, showing a less dense structure, which should give more surface area for increased catalytic activity. Okay, so on our side, the Center for Industry, we're also doing some experiments looking at the catalytic activity of the biochars. So the preliminary work we've done so far was using sawdust as catalyst, and then this was done using methane, which was used to get the first impression of the relative activity of the biochar using non-condensable gases, which are a lot easier to work with than tars until we eventually move over to the tars. And methane is quite relevant to look at because quite often similar catalysts are used for tar and methane reforming. And methane is also an important component in the pyrolysis gas and an intermediate in the tar decomposition reactions. So on the right are some preliminary results, where we did experiments with 10% methane, in the rest nitrogen, but in the second experiments we added also 10% CO2. So in both experiments we see initially quite good conversions for the reaction temperature at 900 degrees, but for the case without CO2 we see a rapid deactivation of the activity as carbon is deposited and reduces the pore space and covers the metallic particles that acts as catalyst. But we see that by adding CO2, the same way as in the activation in previous slide, it helps to gasify the formed carbon. And we see that we can maintain a high activity for much longer time in this case. Okay, so for the last part for the results, we can also mention that we're employing some process modeling here, which is basically simulations that are used to design and optimize the pyrolysis gas conditioning process, where you can see a simplified schematic of the simulations that we're doing on the top right. So for these simulations, currently we're assuming all TARs as a model component just because these pyrolysis gases are very complex. This simplifies the process until we get more data from the experiments. And then we consider toluene and using kinetics from literature to help size the reactor in these simulations. And just as one example from the simulations here, we looked at the conversion of the toluene for different masses of catalysts for different reaction temperatures. And you basically see that to maintain the same conversion, as you decrease the reaction temperature, you would need more catalyst and a bigger reactor. But since the biochar is relatively inexpensive, we've did some preliminary economic assessments that shows that it might be feasible to operate a reactor at relatively low temperatures and then use larger reactors. Okay, so to wrap up, I'll just give a bit of outlook about future activities to take these activities from the lab to a SynoProtein pilot. So one thing that we'll look at is optimizing the activation of the biochar as catalyst, and then also apply this activation to different types of biochar. We'll continue on testing the catalytic activity and compare the different biochar powders, activated pellets, and also benchmark this against metallic catalysts. From these experiments, we'll develop reaction kinetic models for the tar reforming reactions, which can then be used to more accurately size the reactors that we'll need in this process. And then the process modeling will be supplemented with economic assessments and environmental calculations to help optimize the process, to reduce the cost, and to also maximize the carbon utilization in the process. And then lastly, all of these results will be used to design, construct, and demonstrate a pilot of this process. Okay, thank you very much. Feel free to leave your questions in the comments or reach out to me by email if you have any further questions.
Thank you very much for this very interesting presentation on your work. Next, we will have Konstantinos Tendolius from RISE talking about conversion of pyrolysis gas to biohydrogen via gas fermentation.
Thank you, Christina. I will just share. And then presentation mode. Do you see my screen now?
Yes, looks fine.
Yes. Thank you. So my name is Konstantinos, and I work as a researcher at RISE, Research Institutes of Sweden. Today I will talk about the part of the conversion, the biological conversion of the pyrolysis gas for the production of hydrogen. This is a collaboration, a tight collaboration with the University of Borås and RISE. And RISE is the Research Institute of Sweden, the biggest one in Sweden and the fourth of its kind in Europe. And we are mostly located in Sweden, but in other places in the Nordic countries and Northern Europe mainly. We are around 3,300 people working here. And the biggest asset that we have is that we have more than 130 test beds that we can use to help the industry to upscale its processes. Another interesting information is that 50% of our revenue comes from research, and another 50% almost come from the direct industrial projects, assignments. For this group, we are part of, I will just close this. We are a group of people working in this part of the project from the University of Borås, from RISE, and I will choose this one here. The main location where the project activities take place is in Borås, the University of Borås and RISE in Borås, and Uppsala, RISE at Uppsala. And we target a lot the conversion, the utilization of biomass, such as organic biomass, which is produced in vast quantities, large quantities in Europe, such as manure and food waste. For this kind of biomass, there's an established sustainable process that's called biogas production, the anaerobic digestion. And with this process, we can produce fuels and biofertilizer. However, there are also other types of biomass, as it was discussed before in the presentations, which contain lignocellulosic material, the wood and forest residues, for example, or crop residues. And this type of biomass is not easy to biologically degrade because of its complex structure. And here we can use a combination of a thermal treatment, such as pyrolysis, for example. And from pyrolysis, as Henry showed also, we can have different products such as bio-oil and biochar. And the pyrolysis gas we can use as a nutrient in our anaerobic digesters. This is also the core concept of the SynoProtein project, to use low-value lignocellulosic residues and use first a thermal treatment, then a biological treatment in order to produce animal feed and fish feed. From the pyrolysis, the biochar, a part of the biochar will be used, for example, for cattle feed, and the pyrolysis gas will be used as a substrate, as a feedstock in our gas fermentation process. The carbon monoxide from the pyrolysis gas will be converted into hydrogen, and then hydrogen will be converted into single-cell proteins through another biological process. This single-cell protein will be used as fish feed later on. In this presentation, I will focus on the gas fermentation process. And for gas fermentation, we can use different type of gases, such as industrial off-gases, biogenic carbon dioxide, synthesis gas, in this case, pyrolysis gas. And the pyrolysis gas has some main components, which is carbon monoxide, carbon dioxide, hydrogen, and some impurities. Depending on the type of feedstock, on the type of the operational condition in pyrolysis, we have different type of composition in the main components and the impurities. And this is a big advantage when we use a biological process to convert the pyrolysis gases, because the biological process has the ability to use and adapt to different ratios, different compositions of gas components. It is product specific, more product specific in comparison to the thermochemical process, the alternative. However, it has lower yields and lower mass transfer, and this is where we try to improve the efficiency. The main reactants here is the carbon monoxide and carbon dioxide plus hydrogen, which react inside the biocatalysts, which are bacteria. And then we have different products. There is a commercial process for production, for gas fermentation, for the production of ethanol. There is demo processes for production of acids and methane. And we are trying to upscale the hydrogen production right now. The main reaction that we aim at this project is the biological water gas shift reaction. Here is this reaction in 2 steps. Carbon monoxide reacts with 1 molecule of carbon monoxide and 1 molecule of water, react, and we have the production of 1 molecule of carbon dioxide, 2 electrons and 2 protons. Then the protons and the electrons react with, and they form the hydrogen molecule. And from literature, we can see that around 1 kilo of cells, of microbes, can produce approximately 1 kilo of hydrogen per day, depending, of course, on the type of the microbes that we use. For the last year in the SynoProtein project, we started from testing different variables in order to improve the efficiency, the yields, and make the conversion rate faster. And we base our studies in previous projects that we have done. And one of the most important variables is to inhibit the methane formation. because in anaerobic digestion, there are a lot of different series of reactions that happen. Hydrogen is an intermediate product, while methane is the end product. However, we want to stop the reaction at hydrogen production and do not have methane production. For this, we have done a lot of studies on how to do heat pretreatment and how to use chemicals. And another variable is the type of the inoculum. We have used granules, granulated inoculum. We have used suspended cells, and the source of the inoculum, it can be from manure, it can be from digesters or wastewater treatment plants, and so on. Other studies that we have done on this project is to find the best blend for the vitamins that the microbes need to grow and thrive. These are the micro and macronutrients that we have to add, and we will also add a certain amount of carbon source, except carbon monoxide at the beginning of the process, so that we can boost up the growth of the microbes. The gas composition is always very important. It can vary a lot according to, for example, the operational condition of the pyrolysis, the type of the feedstock, and the type of bacteria that we use, the type of microbes that we use have an adaptability on a certain level of variation, of alteration in this gas composition. Other studies that we have done until now is that we have tested with different pH and different temperatures. Especially the temperature affects a lot the cost, the operational cost of the reactor when we talk about bigger scale. And a very important also aspect is the scale up. Whatever we do on these lab scale studies that I talked about these variables, we have always the aim for the upscaling. So whatever we do for these small scale studies we have to be able to do it in the pilot scale reactor later on. And we have worked a lot with mass transfer improvement. So this is our progress until now. First of all, we started at the University of Buros with smaller reactors, these serum flasks, studying all the variables, and then we upscaled in the bubble tank reactor. Then RISE in Uppsala designed and built a trickle bed reactor, which is a prototype that we will use. It is installed at the University of Buros right now. It will be used for continuous carbon monoxide conversion into hydrogen. And here we had to think a lot about the safety environment, which sensors we have to have, which kind of alarms, and which kind of safety aspects we have to take care of. And we plan to design and build a bigger type of reactor. Here is an example from another pilot reactor that RISE has built, and it is active in operation right now. These are 2 trickle bed reactors, and it is a mobile unit, fully automated, and it is used together with a gasification plant. So the plan for the SynoProtein project is to have a similar type of reactor built and operate in 2026, 2028. And this is a little bit more information about the pilot reactor. This pilot reactor is financed by a project, EU project, that is called Biomethanverse. And you see here the gasification unit, and this is the gas fermenter, the automation, the system, the whole system, and the gas fermenters here. And here is a similar concept where wood chips or lignocellulosic residues are used. They are gasified. Then carbon monoxide, hydrogen and carbon dioxide are generated. They are used, this gas is used as a substrate in gas fermentation in these 2 TBRs, the trickle bed reactors. In addition, we have green hydrogen that is added from an electrolyzer, another pilot that is placed nearby, and some vitamins that are added from wastewater. And the target product here is methane, but in our case it will be hydrogen. So from the SynoProtein project until now, right now we have the lab scale reactor, and we're planning to have more experimental work on this. And we have observed that several specific type of inoculum is like the granules give higher hydrogen yields. Also, the higher temperature is beneficial. Then we have also achieved to have only carbon monoxide as the only source of for hydrogen formation. And it is very important to say that stable concentration of pyrolysis gas is essential. However, the small variations can be tackled by the biocatalyst in our TBR reactor. And of course, when we talk about upscaling, we think about the cost a lot. And here we have a lot of observations to take with us for the next step, such as we have protocols that for lower temperature for the heat treatment of the inoculum. And then we have also protocols how to activate, how to store the inoculum, how to adapt the microbes faster, so that we have a shorter startup period. And then we have worked a lot in order to increase the mass transfer of the reaction. And a very important asset here is that we plan to run the lab scale reactor, the TBR in the University of Buros, in parallel with the pilot reactor for troubleshooting, for having lower costs, operational costs towards the end of the project. And in this project, we are part of a very nice group. It's exciting to work with all these interesting partners. And thank you for your attention.
Thank you very much, Konstantinos, for your presentation. Very interesting insights in your work. And yeah, with that, I'm handing over to our last but definitely not least speaker, Biao Li from GTU. Yeah, the floor is yours.
Yeah, now I'm trying to share my slides. Can you see that, Christina?
Yes, I think now you can switch to presentation mode, and then
Now it's okay?
now we see the presenter mode. So I think you can switch display settings.
Let me check.
Also.
What about now?
Yes, that looks perfect. And you are going to talk about gas fermentation for single-cell protein production.
Okay, thank you for the introduction. And my name is Bill Li. I'm a postdoc at DTU. So today I'm going to represent DTU team to introduce our work on this project. And the title is Syngas Fermentation and Single-Cell Protein Production. So we are working at DTU Sustain Department of Environment and Resource Technology, Lyngby campus in Copenhagen. In our research group, we are mainly focusing on combining electrochemical system with other methods, technologies, or platforms such as chemistry, microbiology, material science, and further utilizing this type of platforms or systems for wastewater treatment, resources recovery, CO2 utilization, and other environment applications. So below are our group members. So we are supervised by Ethan, and I'm the one working on this project. And we'll have two postdocs join us next month. So hope we can meet them soon. And then one of the specific research interests of our group is the production of microbioprotein, because this type of microorganisms can produce a biomass rich in protein. So far in our group, we have utilized the yeast, algae, and bacteria for single-cell protein or microbioproteins, because these types of microorganisms can also be combined with the electrochemical system, so to provide a new platform or a technology for microbioprotein production. For example, in here, and we can use water splitting to generate hydrogen and oxygen. Then the hydrogen-oxygen bacteria in the system can use the generated hydrogen and oxygen and the added carbon monoxide and to produce a single-cell protein. I will show more details later. So about the microbioprotein, it has many advantages. So I will just show the bacteria as examples. For example, the missing oxidizing bacteria, MOB, and hydrogen oxidizing bacteria, HOB. These two types of bacteria can be used for single-cell protein. So the advantages of this type of protein, the biomass has a higher protein content, range from 50 to 80%. And it also has a very high growth rate. We can get the highest biomass concentration within several days or even several hours. And also the moderate growing conditions, we can grow this bacteria in room temperature, and also the neutral pH conditions. More importantly, this bacteria has a broad substrate availability, which means that they can not just use some organic compounds like sugars, but also gases, like these two types of bacteria. And also in these generated proteins, they are rich in essential amino acid, which are benefit for animals. And it is independent of length, so which can improve the efficiency compared with the classical agriculture activities. So let's focus on the project. So following the last stage, conversion the carbon monoxide into hydrogen, we are going to develop two fermentation systems to convert the conditioned syngas into a SCP. So the main objectives of this part is to lower the CO2 and hydrogen ratio, enhance the nitrogen utilization and the CO2 utilization to achieve a higher biomass in the end. So we separated this part into 3 parts in the task 2.4. As I mentioned, we will develop 2 systems, type A and type B. So in type A system, MOB, the methane oxidizing bacteria, and the hydrogen oxidation bacteria, HOB, will be inoculated individually. So the MOB can consume methane and oxygen. HOB can consume hydrogen, CO2 from the syngas and nutrient. Then we can obtain the HOB and MOB biomass at the end. In the second system, we mix the MOB and HOB together to use the syngas and nutrient automatically. So finally, we will get a mixed biomass reaching single-cell protein. So specifically, the bacteria will be selected for HOB and LMOB, and the fermenter, the setup, will be also developed and also optimized. So we will also use the synthetic, the syngas, and also the practical syngas from the last stage to feed the bacteria for single-cell protein production. And also the performance of the systems will be evaluated and compared for next step. Then, due to the gas dissolution in the liquid phase is important for gas fermentation, so we are going to develop different bioreactors to enhance the gas-liquid mass transfer efficiency, including classical CSTR, bubble column react, and also membrane reactors based on their different working mechanisms. And second, the high hydrogen demand of the HOB will be decreased to 1 to 7. Then, because we mixed these two types of bacteria, HOB and LMOB, together, so the relationship between these two bacteria will be investigated to see if they are cooperative with each other or competitive with each other. Then in next part, target 2.5, we will try to develop a new system to recover the waste ammonia from wastewater, and also combine with the water electrolysis, or you can call it water splitting, for single-cell production in this system. So here we can see from the right figure, by using water splitting, we can generate the hydrogen and oxygen on the electrode surface, and these gases can be collected. So the nitrogen from wastewater could through the membrane and recovered from the wastewater used as the nitrogen source for the bacteria. So here the bacteria, for example, the HOB can consume the generated oxygen, hydrogen, and also the recovered ammonia as carbon and nutrient for the single-cell production. And after developing this new type of bioelectrochemical system, the effect of ammonium concentration, applied body, pH, and HRT, and on the growth of bacteria will be also studied. And then after that, we will collect the biomass from the electrochemical system to analyze the protein, amino acid, and so on. So in this system, we just achieve in-situ gas generation, utilization, and ammonia recovery for single-cell production. Then in the last part, and we will try to figure out the key challenges in upscaling these bioreactors. So first, for example, for the gas-liquid supply, or as we mentioned, the gas dissolution in the liquid. So in here, we will develop different volume of bioreactors from milliscale to liter scale. And we will also try to see if the high pressure on the reactor will also increase the gas dissolution for SCB production. And of course, the safety issues like the leakage of hydrogen from the bioreactor will also be considered in our lab. And then some operating factors like the inoculum, temperature, pH, and HRT will be also investigated. Due to the syngas also contains some other gases, and also in the nutrient also contains some organic matter. So these types of compounds, the effect of them on the growth of bacteria will also be studied in our lab. And following these 3 targets, we will get a result and a report. So for the design and operation of the system, and the information will be transferred to the next stage. So that's all. Thank you all partners and also the audience. Thank you. So now,
Thank you very much, Biao, for the nice presentation. And I think we are now finished with our presentation. I hope that you learned a lot about our project. I'm now asking all the speakers to turn on their cameras again. And yeah, we are also ready to get your questions if you have any. You can just write them down in the Q&A box that you find in the lower part of the Zoom webinar. So I will give you a few minutes. If you have some questions, you can just post them there. In the meantime, you have already found, I guess, on our website where you signed up for the webinar, but you can also, if you are interested in the project and want to follow us, you can also sign up for our newsletter that you find on the home page of our website if you scroll down. And of course, you can also find us on LinkedIn, and there you can also get all the updates that we have. So let me just quickly check the Q&A if we got any questions. I don't see any questions so far. Don't be shy. You can for sure ask us. But maybe the presentations were all very clear and no questions left open. So there's a question to Hege from Bergenholm. And that is, what is the difference between PSC and PEFC certification?
There, I was struggling with my mute. They are both quite similar. The PFC is more of a group certification, but if one single forest, if it's just the FSC, then you have to certify the exact small or big forest. And the forest structure in Norway is that we have a lot of small forest owners, like farmers. They have a bit of forest, so it will be quite an amount of work because it's a lot of documentation in both certification. So that's why PFC is the most common certification scheme here in Norway. If you see on paper, like for tropical timber, you may see FSC because it's large forest owners, but there are now some, they are getting more and more similar. The PFC is maybe more into the climate effect, and the FSC have some stricter regulation for the biodiversity, but they are getting very similar. And also the Norwegian Forest Standard works with both, and both environmental organizations are involved in both the making of the forestry standards. I'm not sure if that was enough. It's the example is you cannot cut down a whole forest. You need to leave a certain amount of trees. You need to avoid cutting in zones that are close to water. So it's a lot of sort of rules and regulation for how the forest should be managed. And when you are harvesting trees, you need to plant as quickly as possible, and you cannot take out the stumps and the roots and everything. So it's to make sure that that we have sustainable forestry for the future.
Thank you very much for the explanation on that. We have another question that is, so thanks to everyone for the webinar. And the question is how we know, or how everyone can know that the project actually reduces the carbon footprint, and by which method it will be analyzed. I don't know, Long, do you want to comment on that maybe? how we are going to do that in the project?
Yeah, it's an excellent question, and it's the key question also the customers cared about. Just like Hege mentioned, always think about what customers think, and then from the source side, right now has a very good sustainable certified feedstock we use. Then from this part is secured. Then from the whole process, it's a very the key question we are going to investigate in the project as well. And then as I can just touch a little bit on this, I'm not a fully expert on the LCA, but we have the LCA partner as the NOSIS, is the Sustainability Research Institute in Norway, to investigate the whole process. And the research methodology will include both the environmental and social LCA analysis, and which is going to be a very big topic we are going to discuss as well. So we'll have more information, and it's going to be shown in the next webinar as well. So keep posted and in our LinkedIn as well.
Yes, exactly. Thank you so much, Lang, for answering that. So we have, Hege, did you want to add something?
Yes, because
Yeah,
I can maybe add some more information since we do the LCAs for our wooden products. We know that when we use as much bioenergy as we all wood producers use today, we are directly burning the fiber. So for us, it will be a really sustainable choice to sort of save more of that energy into a new product that we can get the right amount of heat into another process. And even this project that saves the syngas. So it's this symbiosis, or what can you call it, like industrial ecology, that we save really the best use of the whole log, because today we are burning a lot of good products for direct heat use. So it's really the saving. So also in our LCAs, we sort of we need to use the mass balance of the whole log, and we put all the customers, all the sort of pieces from the wood need to be calculated into the LCA, and of course this would make a huge difference of saving the biomass more in the whole process.
Thank you for adding that. So the next question is there any difference in HOB and MOB biomass? Guess that's to Biao.
Yeah, I think I can give a short answer for this question. So I think in my side, the main difference between HOB and MOB biomass is the protein content, and also the type of amino acid. For example, for typically for the bacteria, HOB and MOB, when we calculate it and we collect the biomass, we will analyze the amino acid and the protein content. So typically, the protein content would be in the range of 50 to 80 range, but it depends on the bacteria. And then we will analyze the type and also the concentration of amino acid in the protein. For example, the essential some protein or some bacteria contains more essential amino acids, which is beneficial for animal or human. But for some bacteria, they can just generate some more unessential amino acids, which is not good for us. Hope I can. Yeah. Thank you.
Thank you very much. So moving on to the next question. So that is, when you feed pyrolysis gas into an anaerobic reactor, how do you, or are you planning to, make sure that no methanogenesis takes place and the reaction just stops to hydrogen?
Thank you for the question. So we have the pre-treatment, the heat pre-treatment that we apply. and this deactivates or kills the archaea that are responsible for the methanogenesis. So this is how we do it. However, there is also another option of using chemicals, but in our case, we use the best method in order to upscale later on, the most cost-effective one, and we found out that heat treatment is the best. However, why do the methanogens die and the other bacteria do not die? Because the other bacteria can form spores. They are more, they're not so sensitive, so they survive from this pre-treatment.
Thank you very much for taking that question. And the next question is, in which way is the upgrading of biochar catalyst acting in the methane reforming? I think that's to Henry, which is the catalytic function.
Yes, I'm not completely sure if I 100% get the question, so I'll try to answer in the best way I can, but feel free to clarify in case I don't answer it properly. But yeah, so it's the same as I explained for the tar cracking reaction. So it's both the combination of the functional groups and the metallic components in the biochar and the high surface area that also helps that the biochar can be a catalyst for the methane reforming reactions. And then related to the upgrading the activation, it's the same function that you open up the pore surface and increase the surface area for which reactions to take place, which makes it a more effective catalyst. So I do hope that answers the question.
If not, please just maybe post again. Thank you so much, Henry. And then I see another question to Konstantinos again. How do you address the competition between hydrogen production and other products like methane, acetic acids, and so on?
You mean, that is a very nice question, actually, because inside the fermenter, we have various groups of bacteria that can produce different products. However, we can steer the reaction, the direction of the reaction according to the operational conditions. For example, temperature and pH. We can also steer it according to the type of inoculum, the type of the initial bacteria group that we will use. Another way is to use pure strains, pure cultures, monoculture, or specific strains. However, for a long time, for a long duration in the fermentation, we feel like this will be a bit of a challenge to keep the right monoculture without any other bacteria coming in the fermenter. So another way to do it is by having mixed bacteria and enrich with specific monocultures. So all these strategies we try to use.
Thank you so much for giving us those details. So I don't see, another question in the chat that is to Biao. How do you address the safety issues with the gas mixture in the SCP production? For example, explosive gas mixtures. I think you're still mute, or you can unmute your mic.
Oh, sorry.
No worries.
Sorry. That is a really nice question, because recently we also trying to connect other group working on this type of bacteria for the gas fermentation. The important thing is that he mentioned is to notice the gas leakage from the gas bag or gas tank. But now in our group we only use a small volume of gas in a gas bag, and we make sure there was no gas leakage from the gas bag, and we'll put in a fume hood and try to detect the gas in the air, but everything was fine. But we will also try to discuss with other partners about the upscaling for the liquid, especially for the hydrogen in the future? Yeah, that is really a nice question.
Thank you so much.
Yes.
I had someone in the audience raise the hand, but it's gone. So I don't see anyone else raising a question. Also, we are at the end of our time. So I posted you some information where you can find us, also how you can contact us by email in the chat. And then I think I only have to thank you very much for your attention. And yeah, stay posted, join our next webinar again. And then thanks also to all the speakers for the great presentations. Yeah, have a nice afternoon, everyone.
Thank
Bye.
you, everyone.
Bye.